Okay, guys. That was a lovely warm-up. Thank you. Okay. So the exam's hoping to be back by Monday. The Scantron average, and that's not completely like representative, but it's close because it was mostly multiple choice, is a 78. So that's actually good. The thing that's kind of not obvious is that those last 5 questions were 5 points each. So that could destroy things, but so far people are doing pretty well on them. So please stay that way. And I even so far got one person who knew that the Nobel Prize was awarded for vesicular transport on Monday. And I thought that was so fitting. Yeah. Okay, so guys, what I'm hearing is people were confused about the categories of the Nobel Prize. The Nobel Prize that was awarded on Monday was in medicine or physiology. That's a category. So for those who are at medicine or physiology, that'd be a weird extra credit question, right? So, but it's okay. It's okay. Next time, we always have next year for those who want to try it again. Last year was GPCRs, and we're about to learn about GPCRs. So I feel like we're right on the cusp of, I don't know, something. Okay, so this is where we ended. I just want to do like some quick, like just so you don't forget that things go into the mitochondria, because I ran out of time last time. And so just the thing to take away from it is that it's similar to ER, because you've got translocon, you have a receptor, you have a signal sequence. The only difference is you have 2 signal sequences that are reported to the mitochondria. Yes?
I have a question. Is the next test go on the 21st?
I can't tell you that. You'll find out on the 20th. No. So I'm hoping to stick to the schedule, but we have to be, you guys are going to have to be flexible with me. I'm sorry because this is the first year I've gone from 3 times a week to 2 times a week, and so the timing is a little off. And so I'm hoping it's the 21st, but that's soon. So just, I'm going to kind of, as we go into next week, I'll let you guys know. But that's it. I'm sure everyone's wondering that. So there should be 2 signal sequences because you're either going to be imported into the intramembrane space or the matrix. And so you can think of those as 2 addresses for the protein. The protein's imported post-translationally versus post-translationally. So how to ask you to compare and contrast between ER and mitochondria, that would have been one. And then it's similar where there are membrane proteins also in the mitochondria, and they also have these stop transfer sequences and these kind of transmembrane kind of domains also. And then what's happening is they're being imported all the way into the inside of the matrix and then make their way back out depending on where they need to go. Just the basic gist of it. And then porins, these are these channels that are in membranes that allow water and things like that to pass through. So they are these membrane proteins, and they are in the mitochondria. And so they're inserted into the mitochondria outer membrane through 2 steps where they get imported in post-translationally, and they're bound by chaperones, and then they get sent back out through the SAM complex. So they're coming in through the TOM complex, going back out through the SAM complex, and then there are these transmembrane domains, and they move into the mitochondrial membrane, outer membrane. And so just kind of a note of excitingness. The SAM complex is conserved in bacteria and eukaryotic cells, and so this is further evidence, because this is involved in putting these porins into the outer membrane, and this is evidence supporting the endosymbiotic origin of mitochondria. Everyone remember the endosymbiotic theory? Yeah, okay. Mitochondria is a little bacteria. Yeah. Okay. Oh, and then there was this just showing all the different types of signal sequences. And I kind of went over that briefly. Okay, let's move on. So here we are, Unit 2, cell communication, also known as signal transduction. And so most of you guys have had a little bit of a intro to signal transduction in recitation this week, where we started studying the Wnt signaling pathway, which is nice because it's a nice merging from of the two topics that we've been talking about, right? Going from endocytosis in multivesicular bodies and all that into signaling, okay? And the paper is all about how in Wnt signaling, one of the major regulators of beta-catenin stability is sequestered in a multivesicular endosome to stabilize it and allow signaling to take place. And you've seen it in the paper, or you will see it in the paper, and we'll see it in lecture also. What else do I need to say to you? We'll see. Okay, so this is your generic signaling pathway, and I think signal transduction can be as simple or as difficult as you make it. If you're going to focus on every single detail and every name of every protein, every phosphorylation site, you will lose your mind. But I want you to think big picture, okay, as always. You'll have a receptor, and then you've got some kind of thing binding it. And not everything that binds to a receptor is brought into the cell. Sometimes it's just causing the receptor to dimerize and activating it in some way. And then you have some sort of proteins that kind of relay the message, because remember that extracellular signaling molecule is coming from some other cell or from the environment, somewhere from outside, usually from another cell. And so the purpose of it being secreted by another cell is to communicate something to a cell, right? To the cells in the area or whatever. And so you've got all these signaling proteins, which will have all these exciting names, but their purpose is just to relay the message all the way to the nucleus. And what ends up happening is you get somehow activation of some messenger protein that is a transcription factor. And it goes into the nucleus, and it binds to a promoter or an enhancer, recruits RNA polymerase, and then you get gene expression, right? So usually when a cell is communicating to another cell, it's telling the cell to do something. And in order for the cell to do something, it needs to make certain proteins, and that's why this is all happening, okay? Right, so you're facilitating cell-cell communication, relaying a message from outside the cell to the nucleus. The basic end result is to somehow affect gene expression, and then it's involved in cancer because often in cancer, signaling pathways are not working properly, usually due to a mutation, which is causing unregulated signaling. It's all about regulation, meaning is signaling on or off, because that determines whether gene expression is on or off, and if that becomes dysregulated, well, that's bad, right? Because if it's something that's promoting cell growth and it's dysregulated, then you have dysregulated cell growth, and what is that? Cancer, right? So that's the most basic explanation. And I think I want everyone to really think hard about whether they understand this basic big picture. Otherwise, the whole signaling unit is you're gonna want to shoot yourself. Okay? Well, you won't want to shoot yourself, but you'll be really unhappy and confused. Okay? Any questions about the purpose of signaling? Good. Okay, so I'm going to start with a bunch of examples. They're just some of my favorite pathways. And rather than thinking this does this, I want you to think about how they differ and how they're similar in terms of mechanisms of regulation. And they're examples, okay? And as we're going through this whole entire unit on signaling, I want you to think about things in terms of categorizing. There are these main types of receptors. There are these main types of, and they're all different ways of getting a message to the nucleus, but you can think of them as prototypes, and most pathways fall into one of those categories. Okay, so let's talk about the immune response for a minute. So if you're infected with a virus, then one of the things that's going to happen is you're going to get production of interferons by infected cells. And that's their way of saying, there's a virus in the area. And they secrete interferons, alpha, beta, gamma. In this case, we're talking about alpha interferon. And the purpose of secretion of alpha interferon is to go bind to a receptor and let the other on another cell and let the cell know that there's a virus in the area and that needs to respond appropriately. So what would happen is you'll have this one example of one signaling pathway involved in the immune response is the JAK-STAT pathway. And in this pathway, you have your receptor, and then you've got these kinases that are associated. They're bound to the receptors. And when the ligand binds, then you get this cross-phosphorylation, which is activation. And then you get this further phosphorylation, which is more activation. But it also creates docking sites for these STAT proteins. So JAK is the name comes from one of the kinases, which is called JAK. And then this other, this transcription factors that are called STAT. Okay? And so phosphorylation creates this docking site for STAT to bind. And then once they bind, they get further phosphorylated, which allows them to release and bind to each other. So then they form this dimer that travels to the nucleus, binds to a promoter, and activates gene expression. So this is an example of a signaling pathway where you've got a receptor that has kinases bound to it. And the receptors are normally not very close together, but once the ligand binds, it brings them close together and allows those kinases to phosphorylate each other, which allows for recruitment, which allows for activation, which allows for more phosphorylation, which is all just to change the shape of the receptor by this lower phosphorylation, which recruits these other proteins, which then get phosphorylated. So there's all this phosphorylation that you're going to hear about, but I want you to start thinking about what it's doing. And usually it's activating something, inactivating stuff, or creating a binding site for something. Okay? And so in this case, they're binding to each other and going and activating gene expression. Okay. That's the JAK-STAT pathway. So I just told you that activation of the JAK-STAT pathway results in dimerization of STAT, which then goes to the nucleus to activate gene expression. So that was this part down here. Oops. So think about, I want you to put on your thinking cap. Think about import of proteins into the nucleus and what we just learned about. Again, don't forget what we just did in class last week. And I want you to think about what mechanism regulates whether STAT will be nuclear or cytoplasmic, and how does this affect gene expression? And then I want you to design an experiment to the best of your abilities to test your hypothesis. So take a minute to talk it over with your friends. So again, what I'm asking about is how these proteins here are getting into the nucleus. Okay? So what mechanism regulates whether STAT will be nuclear or cytoplasmic, and how does this affect gene expression? Design an experiment. Okay?
One with phosphatase, and the other with so you could say when interacting with kinase, it gets removed, but when phosphate gets removed, it's imported phosphatase or kinase, like when nuclear localization signal
Well, is that happening? Is that happening here? Okay, so I can't wait to hear. I can't wait to hear all your ideas. I can't figure out this thing today. Okay, so the question was activation of JAK-STAT pathway results in dimerization of STAT. That dimerization is required for entry into the nucleus. That might help clarify things. And then it goes into nucleus, activates gene expression. And now, so I wanted you to think about what we learned about import, and then let me know what mechanism regulates whether STAT will be nuclear or cytoplasmic, and how does this affect gene expression, and then design an experiment. So let's start with this. What regulates whether a protein can get into the nucleus? Yes?
Nuclear localization.
So there needs to be an NLS, right? Okay. So what mechanism on this pathway, you can take a look at it here, what mechanisms regulates whether it can get into the nucleus? Well, phosphorylation of what? Because there's a lot of phosphorylation. Yes?
Actually, forms the dimer.
The dimer, exactly. So you need that dimerization. So dimers can enter the nucleus, but monomers cannot. Okay? So if you have a dimer, how does this affect gene expression? It'll turn it on, right? Okay. So I want you to tell me, first of all, what do you think is going on here? How is it getting into the nucleus?
What are dimers? Is it
Dimer means, oh, thank you. Dimer means two proteins binding to each other. Monomer is one protein, polymer is many, trimer is three.
so in this case, it'll be a glucose?
Sorry, oh my lord, what's going on today? So these two, so you have STAT1, STAT2, and that last phosphorylation step allows them to dimerize. And so that's why you've got STAT1, STAT2 dimer. You see that? Yeah. So that's what I mean by dimer. So what's happening here that's allowing STAT1, STAT2 to get into the nucleus when they're dimer, but not when they're monomer?
Is it putting together like a lysine, like I guess chain of lysine and arginine in the dimer?
Okay, so maybe they're creating a lysine arginine rich region. Yep. What else? What is that called? He said they're creating a lysine and arginine rich region when they're dimerized. What is that called? An NLS. Okay, thank you. Okay, so design an experiment to test your hypothesis. So you're thinking maybe they're putting in some kind of NLS that comes from the dimerization. How would you test this? Yes.
Tag it with the NLS and use it, tag it out and see what
Okay, so identify the NLS and remove it or leave it and see where it goes. So mutate lysines and arginines, see where it goes. Okay, that's not a bad idea. What would be your readout? Just looking to see if it was in the nucleus? That's one good one. What other, so let's say you wanted to see like the function of this pathway. So going to the nucleus is one thing, but the function, to test the function, you would have to be looking at what the end result is of the signaling pathway. And what's the end result? Some gene expression. Right, yeah.
So, would we want to like block the enhancer region to then see if it brings that gene?
So do we want to block and enhance the the binding site for the
Yeah.
transcription factor and see if, well, so that's an interesting idea. Block the enhancer region and see if you get gene expression. Well, that really tells you whether or not that enhancer region is responsible for gene expression, but you want to know if you're lysine and arginine rich. So what you want to do is take half of your idea and combine it with his idea and mutate, right? Mutate your lysine and arginine rich regions. Allow them to still dimerize, see if they can get in, and then look for gene expression, right? Because if they can't, one would be, can it get in? Okay, it can get in, it can't get in, but there's a second part, is it has to be in the right conformation to bind to the promoter, which is a whole nother part of it, which you'll learn about in 409. Who's taking 409? Oh, okay. Good. I love seeing faces again. Okay, so good ideas. Anyone want to add to that? Are you guys have your brain moving for signal transduction? The brain shift. Okay, good. So let's talk about the NF-kappa B pathway. This is one of my favorite pathways. I actually work on this pathway. It's fun. Well, I don't know that it's fun. The NF-kappa B pathway is involved in regulation of the inflammatory response. So again, you would have some kind of situation where that brings inflammation into an area. I don't know, like you cut yourself, and then you get all red and hot and swollen. That's inflammation, right? And so those cells in that area are going to secrete. You guys want to share something? Okay, I was hoping you had some great things to talk about. Okay, so the inflammation results in secretion of TNF-alpha by the cells in the area. And that TNF-alpha is going to bind to TNF-alpha receptor, and then you're going to have this signaling. And this signaling looks different than the last one. The last one was phosphorylation, conformational change, nucleus, basically, right? This one is different. So here you have activation of a kinase. So your receptors are going to, the conformation will change upon binding of ligand, and that's going to result in recruitment and activation of a kinase. And then that kinase is going to activate another kinase, which is going to phosphorylate I-kappa B. I is for inhibitor. It's going to phosphorylate inhibitor of kappa B. And that phosphorylation is going to target the inhibitor for proteasomal degradation. When you remove an inhibitor, that allows the transcription factor, NF, nuclear factor, NF kappa B, to go into the nucleus. So again, it's really about allowing a protein into the nucleus, right? So let me ask you a question. What do you think I-kappa B is doing to inhibit NF-kappa B from going into the nucleus? What do you think it's doing? Like, give me a mechanism. You guys want a minute to talk? Let's take a minute to discuss. So let me just reiterate, so I have everyone's attention and you can get the most out of this exercise, right? So this is a pathway dependent on regulated proteolysis. So that means regulated degradation of something, okay? And in this case, the signaling is activating a whole bunch of stuff which ends up phosphorylating an inhibitor, sending it to the proteasome to be degraded, and then the transcription factor is free to enter the nucleus. Huh?
Which part is
I-kappa B is red, NF-kappa B is blue. It's being phosphorylated here, and here it's all broken up because that's it being degraded. And here's NF-kappa B going to the nucleus. So I want you to, yeah. Go. The phosphorylation targets it to the ubiquitin. I was so excited about the proteasome that I threw this at Harris. Nothing against you. Okay, so the phosphorylation targets I-kappa B, the inhibitors of the proteasome. And that allows NF-kappa B to go to the nucleus. And so I want you to tell me what's happening there, okay? Based on what you know about things entering the nucleus. So take a minute, discuss, and we'll reconvene after I put this back together.
Phosphorylate inhibits a transcription factor. Okay. So who can tell me what is I-kappa B doing so that when it's degraded, suddenly NF-kappa B can go into the nucleus? Yes.
It's blocking the NLS.
Yes, it's kind of right up there, but I was hoping you guys would just think about it. Yeah, it's blocking the NLS. There's an NLS on NF-kappa B, and I-kappa B binds to it and obscures it. It's kind of the same thing as when I said you can phosphorylate an NLS and hide it. Well, you can also have proteins that cover up an NLS, and that keeps it in the cytoplasm and prevents it from activating gene expression until all these kinases are activated through signaling through this TNF receptor. And so the NF-kappa B pathway is a crazy pathway, and there's actually tons and tons of receptors that go towards that pathway. So it's not just the TNF-alpha receptor. I just want you to acknowledge the fact that there's many different types of receptors and ligands that activate NF-kappa B. Okay. Any questions about that question? Yes?
Is it correct to just think that the phosphorylation changes conformation in
So for which one are you talking about?
the I-kappa B
Well, so the phosphorylation so the question is it correct to assume phosphorylation changes conformation and results in dissociation? In this case, no, because the mechanism is known, and so actually the phosphorylated I-kappa B is recognized by a ubiquitin ligase, and then it's polyubiquitinated and targeted to the proteasome. But that's actually a good way to think about it, because if you were studying this, you would think many different things, because maybe your readout of your system would be gene expression on or off, And so you might have to come up with your mechanism. And one thing to evaluate would be, is it just not binding anymore? So I think that would be a logical direction to go in. It's just that it's known. Yeah. Okay, good questions. Anyone else? Anyone else? Okay, so what you should be starting to see is a theme of, can the transcription factor get into the nucleus or not? Right? That's the theme here. Okay, so this is an example of a pathway that's dependent on regulated proteolysis, which is similar to Wnt signaling and beta-catenin. But it's a different mechanism, but in both cases, something's either being degraded or not, and that regulates whether something's going to get into the nucleus. Okay? Okay, so this is the Wnt signaling pathway, and you guys have done this already, and we're going to go over it again later. I'll go over it quickly, but the reason I'm putting it up here is because mutations in many signaling pathways are associated with cancer, or they occur in cancer. And in this case, APC, which is this, which is this green guy right here, which is associated with the destruction complex. It's mutated in almost 80% of colon cancers. Hey, who's taking cancer bio? So is this all like merging right now? Isn't that cool? Okay. So just to quickly go through the pathway for those who have not yet had recitation, it will help you probably with thinking about the paper. And for those of you who did, well, you guys can just have a nap right now, or think about it. So in the absence of Wnt, which is an extracellular signaling molecule, you have this constant degradation of beta-catenin because it's interacting with this destruction complex, which contains GSK3, which is a kinase that phosphorylates it, and then it's recognized by ubiquitin proteasome and degraded. And beta-catenin, in this case, is your transcription factor. Okay? So when there's no signaling, the transcription factor is being degraded all the time. It's being made and degraded, made and degraded. But with Wnt signaling, you have Wnt binding to receptors, which then results in disruption of this disruption of the destruction complex. So now GSK3 is recruited to the cytoplasmic tails of the receptors, and it can no longer phosphorylate beta-catenin, so it can no longer be degraded. What it's not showing here is, in fact, it's endocytosed and incorporated into multivesicular bodies. I believe that was not known when this was published, because this was 2008. Maybe it's who got the new version of the textbook. Is there any? I don't even know if we have it on campus yet, but a new version just.
Is it the 5th edition?
No, it's the 6th.
Oh.
I don't know if it's out. I just got an email. I didn't know if it came out for this semester. But I wonder if there will be multivesicular bodies in the new version of the figure. But so this allows beta-catenin to be stable, go into the nucleus, activate gene expression. See the theme? Can it go into the nucleus or not? Is it present? Is there an NOS? Is it degraded? What's going on? Okay. So in this case, you can see how if you have mutations in APC that prevent this destruction complex from working, then you're going to have stable beta-catenin. And beta-catenin is going to go and bind and activate gene expression when it shouldn't be activated. And since this pathway is involved in growth and development and repair, all these kind of like growth processes, you can see how that might be a good thing for the cancer, a bad thing for the cancer patient. Any questions about this concept? And so what you'll see through the whole signaling unit is we'll be talking about different things that can go wrong in cancer. And because almost every single aspect of signaling can be mutated, and suddenly you have dysregulated signaling that can lead to cancer. Yes?
So this pretty much goes either way. Like it's I guess it's bad the fact that the is not being able to the nucleus be expressed, but also
So you're saying it's bad that it can't go to the nucleus, but it's good because you don't want it to go to the nucleus? Well, so don't think of degradation as a bad thing, right? So, because here it's off, there's no signaling, so you don't want it to go to the nucleus. So it's a good thing because it's keeping the beta-catenin activated gene expression off, except for when Wnt is bound to the receptor. Okay? Any other questions? Is anyone napping out there? No, no napping. That's good. Okay. Okay. So again, another example of unregulated signaling in cancer. In CML, a bunch of different leukemias have issues, but in CML, you've got this Philadelphia chromosome, which is where you had this translocation. So there was a breakage in a chromosome that resulted in a translocation, and then you get this BCR-ABL fusion gene, and this results in unregulated tyrosine kinase signaling and affects a bunch of different signaling proteins involved in cell growth, differentiation, apoptosis. So obviously, if you have unregulated cell growth, if you have unregulated apoptosis, because apoptosis is a good thing to prevent cancer, but if suddenly the regulation is not working and maybe you're resistant to apoptosis, that's not good either. So that promotes cancer. And so this is a major, not mutation, a major, I guess, mutation in translocation in leukemia. And so actually, I actually have this drug in my lab right now. If anyone wants some. So I do have the drug in my lab right now, but we're using it for something else, which is very cool. So a drug was developed that's targeting this BCR-ABL fusion protein. So BCR-ABL is this kinase that is always turned on. So its regulatory domain is not there anymore because it's been replaced by this BCR domain. And so you have this constant phosphorylation of substrates, which results in signaling and cell proliferation and survival and leukemia. And so there was a drug developed called Gleevec. Who's heard of Gleevec? Yeah, you guys aren't up to date with all your drug names. So Gleevec actually blocks BCR-ABL, so it can't phosphorylate. It's no longer active. And so it turns off signaling, which helps with treatment of leukemia. And if you catch someone very early in their disease, it works really well. But if they kind of get to this advanced stage, it doesn't work so well. But now there are multiple cocktails. So what happens actually is BCR-ABL ends up mutating further, and it ends up mutating, it's not on purpose, but you get this mutation of the Gleevec binding site, which is not good in this kind of advanced leukemia. But since they made more cocktails, and they have these like multiple drug cocktails they give to people, which deal with all versions of BCR-ABL, and it's fairly successful. So I do have this in my lab right now, and we're using it for something completely different, having nothing to do with this BCR-ABL kinase, but we're using it for breast cancer because it looks like it's upregulating a certain gene that's really interesting to me. So, if you want some, I'll meet you around the corner. Okay. Okay, any questions about that? Okay, so we're going to do a quick signal transduction activity. I've done this in many different formats before, but, well, we'll see. I'm going to try a new one this time. So what I want to do, so there's 1, 2, 3, 4, 5. There's 5 rows of people in this gigantic classroom. And so what we're going to do is, so you're going to be so all the first people in the row, you're going to be the extracellular signaling molecule. And the second people are going to be the extracellular domain of the receptor. Okay, we're doing an activity. Yeah, you look confused. Okay, not you, not you. Are you? Yeah.
Oh, no, yeah.
Okay. It was like- Okay, so here would be your plasma membrane. You guys all in the middle are signaling proteins. And then somewhere over here, we're going to have transcription factors. So you're going to be a transcription factor. You're going to be a transcription factor. You'll be a transcription factor. You're a transcription factor. And you're a transcription factor. Okay? And so you guys are the nucleus. And so if someone needs to be a promoter, who wants to be a promoter? Okay, you're a promoter, 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 yeah, you. And so then back here you guys are going to be RNA, okay? mRNA. We're going to skip processing. Okay, so so this is what signaling is, right? Because what we're going to do is it's going to be like telephone, okay? Okay. Who played telephone before? Does anyone not know what telephone is? International people, are we good on telephone? Okay. I understand there are things that are very American, like Wizard of Oz. Don't ask Chinese adults about Wizard of Oz. China was closed then. Okay. Anyway, I'm just saying, right? Who's Chinese? Chinese, no. It's true. Cultural bias. Okay. Anyway, so what I'm gonna do with this craziness here is I'm gonna tell you each receptor, each signaling molecule. Each signaling molecule is gonna have a message, and then I'm gonna make sure it's correct in the resulting RNA. Okay, and if it's not, that means you're cancer. Okay. Okay. Ready?
Oh, we are so good.
Okay, does everyone have their RNA? Okay, what do you have? Donuts?
Applesauce?
Applesauce? Ice cream? Soup? Information? It's inflammation. Where's the mutant? Okay. So that was more than just a fun thing to do. That is what signaling is. And somewhere along the way in the back row, something was dysregulated. And we got information instead of inflammation. But basically, something was secreted by a cell, transmitted all the way to the nucleus, and then you've got your RNA out the other end. And hopefully it's donuts. Okay, so everyone understands the big picture, right? Great. Okay. So this is just background stuff. So cells are communicating through binding of receptors by signaling molecules that are secreted by other cells. So signaling molecules, there can be different kinds. You can have hydrophilic signaling molecules. that bind cell surface receptors, right? They're hydrophilic. They're not going to want to pass through a plasma membrane, so they're going to bind to a receptor. There could be small hydrophobic signaling molecules that are going to diffuse across a membrane and bind to intracellular receptors, often nuclear receptors. So those are different kinds of signaling molecules. So signaling molecules can remain bound to the signaling cell or secreted. They can act over short or long distances. So you can have this contact-dependent signaling. This is like what we talked about with the CD4 HIV paper we did, where I was like, remember, CD4 is not bringing anything into the cells, binding to something in another cell. So this is where you've got these cells that are communicating because there's two kind of membrane-bound receptors interacting. You can have this paracrine signaling, where you have a signaling cell that's secreting some kind of molecule, and it's binding to receptors locally on target cells. You can have synaptic. Okay, so I'm gonna admit something to you guys. I have never in my life taken a neuroscience class. So anything, I know, it's tragic. I also don't know anything about organismal anything or ecology, but I respect all. But so whenever anything comes up, I'm gonna go hand waving because I'm not gonna try to tell you I know anything. But there is synaptic signaling, which you guys learn about in physiology, so it's fine. And that's neurotransmitters. And then there's endocrine, so that would be hormones. What?
It's overlapping.
It's like we're taking animal fins and we go.
Who's taking animal fizz? Oh yeah, I love when things all come together. Okay, so hormones. Remember, hormones are secreted. They go through the blood. They go throughout the whole body. And hormones tend to be hydrophobic or hydrophilic. Hydrophobic, so that it diffuse across membranes and stuff like that, and get into target cells, and usually bind these nuclear, not all of them, but a lot of them. Usually bind to nuclear receptors, but there's also cell surface receptors for some of them. Good. Okay. Different cell types respond to different combinations of signals, and that affects the behavior of the cell. You can get signaling that causes cell survival. You can get signaling that promotes proliferation, so cell division. You can get signaling that promotes differentiation. That's one of the ones that Wnt signaling does. And then you can get signaling that causes the cell to kill itself and causes apoptosis. And then the same signaling in different cell types can cause completely different things also.
How does Wnt cause differentiation?
Wnt is involved in development. So beta-catenin is going to go to the nucleus and bind to receptor promoters and activate gene expression. The genes that are being activated tend to be involved in development and differentiation. Yep. Okay. We're good. So different cell types respond differently to the same signaling molecules. That's what I just said. Heart muscle, acetylcholine might cause a decreased rate and force of contraction. That same neurotransmitter in a salivary gland cell might cause secretion, and in skeletal muscle, it might cause contraction. Same signaling molecule, different outcome. Why do you think that is? So you've got a signaling molecule, you have a receptor, but it's that in-between stuff that's different. Yeah. It activates different things. But so let's think even more deeply about that. So what do you think is different that's being activated?
The actual receptor.
Well, the receptor is going to be the same because, well, I'm pretty sure the receptor is going to be the same. Where did I hear kinases? So some kinases, so some of the intracellular signaling molecules.
Like each of the cells form different mechanisms, or they have different effects over what it actually changes in the cell. Is that right?
Right, so each cell has a different job, so they're gonna have different things that are happening, so different gene expression, yes?
When each cell, like skeletal compared to like maybe something like, I don't know, something that's in your intestinal cells, don't they have, even though they have the same things in them, but there's different amounts of things in them.
Okay, different cell types have the same things inside, but different amounts of things, so, so you mean transcription levels change in different cell types? Yep.
Even though the receptor's the same, the cytoplasm can be a little bit different.
Oh.
So that way it can receive the same molecule, but it's in different messages.
If the receptor's the same, can the cytoplasmic tail be different? I am not sure about that because my instinct is to say no, it's further down the line. But there are splice variants of different genes, and it's conceivable that there might be alternatively spliced receptors in different cells that might somehow affect that. It's a logical thought. Yeah. Anything else? Right, so it really all has to do with the number of not transcription factors, but these signaling molecules that are going to get that transcription factor into the nucleus, and the transcription factors that are activated by, because one signaling molecule, well, let me take that back. It has to do with the number of signaling molecules, and it also has to do with the genome and that epigenetic regulation of the genome, which we're not really talking about in this class, but seeming important. Basically, some genes are going to be highly repressed in certain cell types because they're not needed, so they're not going to be activated. Okay, good. So cells communicate through binding. Did I already do that? No, do I have a repeat? I have a repeat. Okay, so some signaling molecules diffuse across the plasma membrane and bind receptors inside the cell. Right, so this would be nitric oxide, steroid hormones, thyroid hormones, retinoids, vitamin D. Would you say these are hydrophilic or hydrophobic? Hydrophobic. Because a hydrophobic molecule could diffuse across the membrane, but a hydrophilic molecule really does not want to. It does not want to, because remember, I see a confused look back there. Because a plasma membrane is hydrophilic or hydrophobic. I mean, on the there's but yeah. So there is the hydrophilic heads, the polar heads, but really to get through there, it's fairly hydrophobic in the center. So you're not going to be able to just make your way through. Okay. So here's one example of something. So nitric oxide gas produced by endothelial cells in blood vessels diffuses across smooth muscle cell walls to stimulate muscle relaxation. So you have your nerve cell, and it's releasing acetylcholine, which is going to bind to receptors on endothelial cells and activate production of nitric oxide, which will diffuse across and cause that muscle relaxation in the smooth muscle cells, right? And so here's another way to look at it. But this is what's causing smooth muscle cell relaxation. So here's your nerve cell and your acetylcholine, and you have binding to a receptor, which results in nitric oxide production in the endothelial cell, and it diffuses across membranes and binds to and activates guanyl cyclase, which makes cyclic GMP, which results in rapid relaxation of smooth muscle cells. That's just how we relax, okay? So nitroglycerin is used to treat angina, and it is converted to nitric oxide gas, and so that's how it induces relaxation of heart muscle, and that reduces the workload of the heart. And then Viagra inhibits cyclic GMP over here, cyclic GMP phosphodiesterase, so it's not broken down, and so it lasts longer. And so it keeps the relaxation of the smooth muscle cells longer. Does anyone know what Viagra does? No, I'm just kidding. It's on YouTube. Okay. Okay. So small, so small hydrophobic signaling molecules, they bind to nuclear receptors and stimulate gene expression. So I just want to say about 10 times right now, nuclear receptors, because it's called receptor, people always think it's on the cell surface or the surface of the nucleus or the surface of something. It's not. It's a protein that shuttles back and forth between the cytoplasm and the nucleus, and it, when it's bound to its hormone, it's in a conformation that allows it into the nucleus to activate and bind to the promoter to activate gene expression. And so I'm just saying this now because on the exam, it'll have some kind of question like it was on the quiz about where you'll find nuclear receptors, and you're gonna all tell me it's on the surface of the nucleus, and I'm gonna say wrong. So don't do that. Okay, I bet you I need new batteries because there's another delay. Okay, so here's your nuclear receptor, and it's just a protein. And it has different domains. And so remember, we talked about the concept of domains, and here's an example of different domains in a protein. You've got this protein, and it has this shape, and it'll have a transcription activating domain. It'll have a DNA binding domain. It'll have a ligand binding domain. And then it has this domain that will bind to an inhibitory protein. And so in the absence of the ligand, which is, let's call it some kind of hormone, in the absence of the ligand, it's in this inactive conformation. But when you have the ligand present, there's a gigantic conformational change that kicks off this inhibitor and recruits a coactivator and allows binding to the DNA and then activation of gene expression. So that's how that works. In this case, the domains just mean the parts of the protein that do a certain thing. So a DNA binding domain is the part of the protein that binds to DNA. It's not a different protein. It's part of that same protein. Okay? Any questions about this? Yeah. Okay, so we've got 3 classes of cell surface receptors that we're going to start talking about. We've got ion channel coupled receptors, G protein coupled receptors, and enzyme coupled receptors. So what would you say JAK STAT pathway, the receptor there, what category does that fall into? Enzyme coupled. Yay, good job. Good. Nuclear receptors are not located on the cell surface. I'm saying it here again, so you'll see it again and again, and it'll be drilled in your head. You guys get to benefit from mistakes of past classes. Isn't that nice? In like 10 years, I'm just going to be giving out test questions. Right? I'm just like, don't get this wrong. This will be on the test. Don't get it wrong. G protein coupled receptors. What can you tell me about G protein coupled receptors that you might notice from G proteins or GTP binding proteins? I'm just wondering if you can link a few things together. G proteins. Anyone come across anything? Yes. GPCRs. Give me more. Tell me more. Okay. What do G proteins likely bind to? GTP. How does that affect their function? It regulates whether it's on or off in some way. So going back to RAB and RAN, and we're going to have more, but like there's this theme of GTP binding proteins, and GPCRs, G protein coupled receptors, are GTP binding proteins, but that are receptors. And we'll learn more about that. Okay, so we've got ion channel coupled receptors. So here, the signaling induces a conformational change affecting the ion channel, so opening or closing the ion channel. This is involved in rapid synaptic signaling between nerve cells and electrically excitable target cells. These can be regulated by GPCRs. I don't do too much about this one. GPCRs, they have this inactive receptor and an inactive G protein, and you'll get signaling which will cause a conformational change and activation, which will activate this another membrane protein. So they actually activate other membrane-bound proteins indirectly. And we'll learn more about that. Yeah? Yeah? No? Okay. And I used to always be like this in class or something like that, and I used to always get called over. The worst, though, was when I yawned loudly. Did I tell you guys that story yet? I was so out of it. Like, you know, I think all undergrads have a right to just be out of it because you're juggling all these different things. And I was so out of it, juggling my million hours of work per week and all this classes. And I sat there in class and I was just like, it was horrible. I didn't even know I did it. The professor was like, wow. You know, if you're bored, you don't need to be so loud about it. And I was like, huh? I didn't even know. And then 5 seconds later, I was like, that was me. So don't yawn loudly. Luckily, it was dark because those were the days of overheads. And so the whole class is dark, which makes you even more tired, right? The whole class is dark except for like a spot like in front of me. So I'm completely falling asleep. And he didn't see me. I don't think he saw me, and he was me. Funny thing is that he came to visit Towson last year, the same professor. And I'm like, hey, I took your micro class in like 19, I don't know, some long time ago. And he was like, oh, I think I remember you. Like, mind you, it was a class of like 250 people. But I didn't say I was the one who yawned loudly.
He would have remembered you.
He probably would have,
He would have
right? Right. Anyways, that's my story. It has nothing to do with GPCR. Enzyme coupled receptors have intrinsic or associated. Now I want you to understand this. Intrinsic or associated enzyme activity. What does intrinsic mean to you? I'm gonna use my class list that I brought. That way I can find out who's not here. Stephanie. I know that happens sometimes.
Like something
Intrinsic. Huh?
Intrinsic
Inside. So, okay. So if you have intrinsic, if a receptor has intrinsic enzyme activity, that means it has its own enzyme activity in its own protein. There's no associated enzyme. It has a domain. It usually has a kinase domain. Okay? And associated is like with JAK-STAT, where it actually had enzymes bound to it. Now let's go back to that CD4 paper that we did last week, and we were talking about CD4 and this P56. That's kind of like an enzyme associated with a receptor. Okay. So usually you'll have. So this is actually showing the intrinsic enzyme activity. Notice there's no additional protein bound, but you'll have a signaling molecule. In this case, it's in the form of a dimer that will bind to the receptor and bring them close together, and then they can phosphorylate each other. Or sometimes what you'll have is the signaling molecule will be associated with an enzyme, and so the receptor will be associated with an enzyme. So the signaling molecule will then bring two parts of the receptor together and allow for enzyme activity to occur. Usually phosphorylation of cytoplasmic tails. I think I might stop here. You guys can go.
How does it bring it together? It's like, is it pulls it through the membrane?
I'll tell you in just a second.